Hello, so this is the week 4 enzymes lab, and it's on Valentine's Day it's due. Oh my gosh. Oh well, sorry for that, you guys. Well, I'm not having much of a Valentine's Day, so I will torture you all. Husband's just busy. Okay, so this is what you're gonna see. The readings all listed on the syllabus, right? You want to see my faculty documents, you'll get the formatted one. It looks the same, it's just not ugly. And then you also have the rubric. So let's go ahead and look at the rubric first because that just seems to be making me happy today. Okay, so the rubric, we have 3 exercises, and the third exercise is going to include the laboratory summary for points. So it's probably really short, and then 0.75 for all the complete sentence and grammar, and don't do weird highlighting, have in-text citations if you need it. Really the big thing is complete sentence, write it like you meant it, not like you half-assed it. Okay, not like you were in a rush and did it the last minute. For this one, it is on enzymes, so to me it's a little bit more stressful just because you're like not used to dealing with it, but it ends up being a lot more interesting in the same point. So let's go through it. Give me a second to... Okay, so you'll put your title page in as always. And scroll down. Observations, you can do blue or black, light blue, and amber. That is the color. You'll just put a plus. A plus if there's nothing, it's a negative. And then you will do that in cross time, so give yourself time to do this. Really for labs, give yourself probably 3 times as much time as you think it's going to take, because they just, I don't know. For me, labs are always very stressful, if you haven't figured that out from my various videos. They're fun. You learn so much, but they're so stressful. So you have the different temperatures, and you'll do it, and you will see stuff happening at different times. Basically, you'll see, right, a lot of starch, and then it should go down to no across time, right, because you'll be processing it. We'll go through that, and then we go into here. Did I skip one? What are we on? Table 2. Show work. That's it for this part here. Show the work. Okay, so just say whatever divided by whatever or da. You can just say whatever divided by whatever equals your percentage, and that's just mostly because if you get this part right but you get the number wrong, like at least I knew you were heading in the right area, and I can just deduct. I probably won't deduct points. I don't know. Can you not do the math? Should I deduct points for not doing the math? I'll be probably pretty gentle if this is correct and the actual answer is incorrect, okay? And then this is the same thing. You're processing starch again. So you'll probably see everybody have starch, and then it will slowly die off with probably one of the test tubes never changing, which would be your, what you may call it. All I can think of is sample. The thing that has nothing in it. I will think of it later. This is why you should not make videos early in the morning. Your control. It's probably your control. See, shows up eventually. Okay, so then 3, you're going to do the same thing, whether starch is there or not. You'll put in your pHs this time. That's the variable, obviously. I don't know if it's obvious, but the first time it was temperature, right? This time it's concentration, and this time it's going to be pH. So that's what you're really looking at, like across the pH, what happens to your enzyme. And you'll see the same thing where pretty quickly it's going to go away, or maybe it doesn't go away at all because it's too acidic or too basic, right? And then we have some questions. Remember, as always, you can submit drafts of any of these. I think I forgot to say that on the other video. Again, why you should not make videos before coffee. Or caffeine. I don't actually drink coffee, but caffeine. Caffeine is a beautiful thing. Alright, so we want to know what's the color if there's still starch, what's the color if there's no longer starch, when it disappeared, what happened, probably got eaten up or something, what's the variable, so this you want to make sure you're doing the temperature one, so probably the temperature, what's your control treatment, probably the one where nothing happened. So based on the data table, blah, blah, blah. So where's it doing the best? What's the best, right? Let's check all of that. 37, yeah. All there. What's the second best? And where did nothing happen? Why do you think nothing happened? Guessing was denaturing was involved. And then chosen as mid-range. So why is 37 mid-range? It wants you to convert C to F so you see what it is in a way that you understand it. And what is that temperature really like? So when you convert this, it's going to be a temperature that's really, really familiar to you, and that's probably important. Why is it important to immediately observe the color of the test tube contents? Why didn't you add an iodine indicator directly to the test tubes? And what happened when you incubated them? Okay? What happened after you added a mil, or cm, sorry, I don't know where mil came from. Why did this occur after the second addition and not before? So, for J, be thinking, we probably, we made them, they were hot and they got cooler, right? So maybe that has something to do with it. And then construct a graph. So this is how you want to do it. You're probably going to do a bar graph, would be my guess. Just come up with a graph. The example is a bar graph with temperature up the left side and on the bottom time. And then test tube 1, 2, and 3 up that left column and across. If you need help, let me know, and I can give you at least what the bars, what the axes are for the graph. And again, this is something where I would probably draw it and stick a picture in, because otherwise it's too much work. You can do it in Excel and port it over, or you can do it, I don't think you can well, you could really do it in Word, but it would be a pain in the butt. But generally, just draw it. It's fine. Whatever's easiest for you. Do what's easiest for you when it comes to that stuff. Because this lab is not learning, lab is about enzymes, right? Not learning how to get stuff from Excel into Word. So do what's easiest, whatever makes sense to you. But don't skip it, because it'd be sad. No, do it. Okay, discussion, just make sure it's in the lab. Okay, discussion, what was the initial hypothesis? What have you learned? What was practical? Okay, so now we're going to talk about substrate concentration. So you know it's probably what they're going to ask you about somewhere. And then say, what's the variable? What was the variable? Probably the concentration of your substrate. Where was it the highest? Concentration in the starch. And then how could you improve this experiment? I always like that. How could you improve? And then make a pretty chart. Same thing. So does it make one? This is G. That helps if you're in the right spot. They're doing the same thing for this one with a bar graph where across the bottom is time and across the left is your test tubes, okay? So that's totally fine and pretty simple to do. It tells you what it is. I don't know. Pretty simple. You can do it. You can do it. Okay, was it supported or refuted? What have you learned? What practical application? So what industry might this help in? And then describe another experiment that could test the same concept. So then we have enzymes and pH. So what's our variable? Probably the pH. And then where is it the best? Where is it the worst? Why? So here, when you do the why, you're probably going to be wondering if it's too acidic or too basic. Would be my guess. Did the result refute? And then laboratory summary, just how you might propose, what you learned, practical applications. I do love the practical applications. They just make me happy. Don't they make you happy? Okay, so as you can see, the lab's not too bad for the work that you're going to be doing. It's just the actual playing with your enzymes at the different pHs. So as I said, if you start to feel nervous on it, just take a deep breath. And you can do it. It's not going to be too bad. I love to review a draft, so if something went wonky, I can tell you what you should have seen. Also, feel free to share your observation tables so you can see if something went wonky, because if we have a bunch of students and you're seeing everybody kind of got one result and you got a different result, then maybe something went wrong and maybe you could explore that, which is something that I would tell you if I was reviewing it. I'd be like, well, your data really should have shown that this pH didn't have anything. So I would just say, like, as a note, say, like, I should have seen this, but maybe my pH indicator was old or something, okay? So we'll we'll work together if that happens, or if you need help. Just, reach out to me, reach out to the other students. Let's try to do this all together so we can get this done and do it right and have fun. Like, it's going to be really fun to see the color changes and play, and you're going to really feel like a scientist. So try to embrace that. Okay, bye.